Hello dear students. In the chapter biomolecules, we have already studied about various types of biomolecules till now. We started with the proteins, then we studied the different types of lipids, and in last class we studied about the carbohydrates. Today we will study about the most interesting type of biomolecules which are called as nucleic acids. What are nucleic acids and why are they called nucleic acids? First, the nucleic acids are the combination of DNA and RNA. You know the full form of DNA? DNA stands for deoxyribonucleic acid and RNA stands for ribose nucleic acid. Okay, so both of them together, they are nucleic acids. Why are they called nucleic acids? Because they were found in the nucleus of the cells by the scientist, that is Friedrich Mischer, who called them as nuclein. Later on, they were considered as nucleic acid because of their acidic nature. So, because they are present in nucleus, that's why they are called as nucleic acids. But actually, they are big molecules. They are polymers and they are called as polynucleotides. They are the polymers of nucleotides. And these nucleotides are held together by special types of bonds. And these bonds are called phosphodiester bonds. What are phosphodiester bonds? We will study the details of the phosphodiester bonds. But before coming to the phosphodiester bonds, we must know what is the meaning of nucleotide. What are nucleotides? Nucleotides are also big molecules which are made up of nucleosides and phosphoric acid. When a nucleoside is held together by an ester bond and attached to a phosphoric acid, then the structure is called as nucleotide. Okay. ATP is a type of nucleotide. We'll study about it later on. Okay. But what are nucleosides? Nucleosides are also big molecules which are the combinations of a specific type of pentose sugar along with nitrogen bases. And these pentose sugar and nitrogen bases, they are held together by glycosidic bonds. These glycosidic bonds, they hold one pentose sugar and one phosphoric acid. One nitrogen base. So these pentose sugars, we have already studied about the pentose sugars. They are ribose and deoxyribose. You know the structure of ribose and deoxyribose. Ribose structure is like this. So it is a pentose sugar having this structure. This is ribose. The formula is C5H10O5. Last time we studied. Likewise, deoxyribose is similar, having the formula C5H10O4. So the structure is slightly different only with one hydroxyl group. Instead of one hydroxyl group, here is only H. Otherwise, the structure is same. So these are ribose and deoxyribose. When these ribose and deoxyribose, they are connected with a glycosidic bond, then the structure is called as a nucleoside. We will study about different types of nucleoside. But for studying that, we must know what are nitrogen bases. Nitrogen bases are again the biomolecules which are the ring structures. And these ring structures may be of two types. Pyrimidines having a single ring and purines having two rings. This is a single ring made up of 6 atoms, 6 carbon atoms. Whereas here 6-membered ring is there. Here there are 2 rings. One is 6-membered ring, other is 5-membered ring. So such structures are called purines. When only a single 6-membered ring is there, then that ring is called as, that base is called as pyrimidine. Why are they called nitrogen bases? Because nitrogen is the integral part of the ring. That's why they are called nitrogen bases. Here also you can see. Okay. So the examples of pyrimidine, there are only 3 types of pyrimidine. Uracil. This is the structure of uracil. You must learn this structure. It is given in your book also. So uracil. Likewise, there is another nitrogen base, thymine. In RNA, it is uracil, whereas in case of DNA, it is thymine. So thymine and uracil are different. Almost they are similar, but in RNA, it is uracil. In DNA, it is thymine. So this is one type of pyrimidine. Also, cytosine. Cytosine is present both in RNA and DNA. Now, when we come to purines, purines are 2 in number. There are 2 types. One are called adenine. This is the example of adenine. When we say ATP, adenosine triphosphate, so that nitrogen base is adenine. There are 2 rings. This is a 6-membered ring and this is a 5-membered ring. This is 6-membered ring. Examples are adenine and guanine. So the basic property of the nucleosides and nucleotides is with respect to the nitrogen bases, because all other things will be same, but the nitrogen bases will be different. So based on the type of nitrogen bases, there may be nucleosides and nucleotides. Now we will study about the different types of nitrogen bases, their nucleosides and their nucleotides. Now let's see the difference between a nitrogen base, a nucleoside and a nucleotide. Many questions are asked based on this. In NEET exam also, you may be given a sequence and you may be asked to figure out the odd one out. So you must know which word stands for a nitrogen base, which is a nucleoside and which is a nucleotide. Let's find out. First, if we take uracil, uracil is a type of nitrogen base. And when there is a sugar attached to it, suppose pentose sugar, then it will become uridine. And when a phosphoric group is also attached, then it will be called as uridylic acid. So uracil is a nitrogen base, uridine is a nucleoside, and uridylic acid is a nucleotide. So you must know. When if I ask you, this is in case of RNA. This will be present in case of RNA. If a question is asked, what will be there in case of DNA? You will say, in DNA, there is no uracil. So such structures will not be there in DNA. Neither uracil, uridine or uridylic acid. Fine. Now in DNA, instead of uracil, the nitrogen base is thymine. So thymine is a nitrogen base, whereas thymidine is a nucleoside and thymidyllic acid is a nucleotide. So if you are asked what is the difference between the components of thymidine and thymidyllic acid, what will be your answer? In thymidyllic acid, there will be a phosphate group attached, whereas here only nitrogen base and the sugar, pentose sugar. Okay. So if you are asked which will be the whether this thiamidylic acid will be present in RNA or DNA, it will be only in DNA, because in RNA thiamidylic acid will not be there. Okay. Now next is cytosine. If the nitrogen base is cytosine, then the nucleoside will be cytidine and the nucleotide will be cytidylic acid. Likewise, if you see the nitrogen base is adenine, it will be adenosine, nitrogen nucleoside, and adenylic acid will be the nucleotide. When there is guanine, it will be guanosine and guanylic acid. So this was the complete chart showing nitrogen bases, then nucleosides and nucleotides. Now we will study the most famous structure of DNA, that is double helical structure of DNA, which was explained by Watson and Crick. Let's find out and study the structure of DNA. Now let's study the most famous double helical model of DNA, which was proposed by Watson and Crick, for which they got the Nobel Prize also. So this double helical model of DNA has got two chains of polynucleotides having ribose as the pentose sugar. So these two chains were anti-parallel. Anti-parallel means the 3' end is top here and 5' end is top here. Okay, so we will study a small part of this polynucleotide, then you will understand better. So before understanding this, let us first know what is this double helical model of DNA, what they explained. They explained that there is a complementary base pairing between the nitrogen bases. Like wherever there is adenine in one strand, there will be thymine complementary to it on another strand, and they will be held together by two hydrogen bonds. And wherever there will be cytosine, another chain will have guanine, and they will be held together by three hydrogen bonds. Fine. So let us study a small part of one DNA. So if we take 4 nitrogen bases on one side, what will we see? Fine, let's see. Suppose this is one pentose sugar. This is another pentose sugar. Third pentose sugar. Fourth pentose sugar on one side. So you know this is anti-parallel. There will also be a pentose sugar, but it will be in opposite direction. One here, two here, three here, four here. Now this 3 and 5, what does it mean? This means that the position of carbon is 1, 2, 3, 4, and there is the 5th position here. So on this position there will be one OH group. Likewise, if you see, the same goes here also. So this will be 5 prime, this will be 3 prime. 5, 3, 5 prime, 3 prime. Understood? So same way it will be like this here. Here it will be 1, 2, 3, 4 and 5 will be here. This will be 5 prime and this will be 3 prime. It will be OH here. The same goes for this also. So this will be 3 prime, this will be 5 prime. 3 prime, 5 prime. 3 prime, 5 prime. The polarity is very important in nucleic acid. Now, this is one pentose sugar, one deoxyribose, to which suppose there is any nitrogen base attached. Suppose here the nitrogen base attached is adenine. Why I have shown this? It is a two-ring structure. So on this side, the nitrogen base attached will be always thymine, and they will be held together by two hydrogen bonds. Let's see here. Suppose here it is cytosine. Here the nitrogen base will be guanine. This is pyrimidine, this is purine, and they will be held together by three hydrogen bonds. Suppose here the nitrogen base attached is thymine. What will be here? It will be adenine, and they will be held together by two hydrogen bonds. Suppose here it is guanine. The nitrogen base attached here will be cytosine, and they will be held together by three hydrogen bonds. So you can see in this way this polynucleotide is attached, and now this bond which is present in between this is a glycosidic bond. This is also a glycosidic bond. This is also glycosidic. This is a glycosidic. Now they are not attached till here. At the fifth position, now it will attach with a phosphate group. Suppose a phosphate group is attached at this position here. Suppose a phosphate group attached here is attached here. There is one phosphate group attached, one phosphate group attached, one phosphate group attached here, one phosphate group attached. Likewise, one phosphate group will be attached here, 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 here. And these phosphate groups will be held together by ester bonds. So the bond formation will be in this way. Bond will be formed here. Bond will be formed in this way. Likewise, let's put it in the circle to make it more clear. So they are held together by phosphodiester bond. So you can see the topmost position here is 5 prime, whereas the topmost position here is 3 prime. Here on 5 prime there will be always a free phosphate group, and on the other chain the 3 prime end will be a free hydroxyl group. Likewise, here there will be a free hydroxyl group here, 3 hydroxyl group here, and here a free phosphate group will be there. So this is the structure of a DNA. You will also study the similar structure and more details of the complementary base pairing in class 12th genetics. Okay, so this was about the biomolecule nucleic acids. So nucleic acids are the big biomolecules and they are held together by phosphodiester bond. So we have already studied about different types of biomolecules. So in the next lecture, we'll study about the enzymes. I hope you have understood the structure of the polynucleotides, that is nucleic acid, DNA and RNA. The basic difference between DNA and RNA is that DNA is a double helical structure. There are 2 chains, whereas in case of RNA, it will be only a single chain. Other difference would be the pentose sugar present in DNA will be deoxyribose, whereas in case of RNA, it will be ribose only. And When we talk about the nitrogen bases present in DNA will be ATCG, adenine, thymine, cytosine and guanine. Whereas in case of RNA, it will be AUCG, adenine, uracil, cytosine and guanine. Okay, I hope you have understood these polynucleotides, that is DNA and RNA. Thank you very much.